June 26, 2000. The East Room of the White House. President Bill Clinton stands between two scientists connected by video, Francis Collins in Washington, Craig Venter in Maryland. They've been bitter rivals in a race to sequence the human genome, but today they're declaring a truce. Clinton announces, "Today, we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, the wonder of God's most divine and sacred gift." Regardless of one's theology, the moment is historic. After a decade of work and billions of dollars, scientists have produced a working draft of the human genome, all 3 billion letters of DNA that make a person. It's biology's equivalent of landing on the moon, a milestone that will transform medicine, reshape our understanding of humanity, and raise profound ethical questions we're still grappling with today. This is the story of the most ambitious biological project ever attempted, how scientists mapped the human genetic code, how public science competed with private enterprise, how international collaboration achieved the impossible, and how reading our DNA is changing what it means to be human. To understand the Human Genome Project's significance, we must first understand DNA and what a genome is. DNA, deoxyribonucleic acid, is life's instruction manual. Every cell in your body contains the same DNA, a molecule shaped like a twisted ladder, the famous double helix discovered by Watson and Crick in 1953. DNA consists of 4 chemical bases, adenine A, thymine T, guanine G, and cytosine C, arranged in sequences along 2 strands. A always pairs with T, G with C. These bases are the alphabet. Their sequence is the code. Groups of bases form genes, which provide instructions for making proteins, the molecules that do almost everything in your body. Your genome is your complete set of DNA, about 3 billion base pairs in humans, divided among 23 pairs of chromosomes. But in 1985, when the Human Genome Project was first seriously proposed, sequencing the entire human genome seemed absurdly ambitious. DNA sequencing technology was slow and expensive. The largest genome sequenced was a virus with 170,000 base pairs. The human genome was 3 billion base pairs, 20,000 times larger. Many scientists thought the project was impossible or wasteful. The idea emerged from several sources. In 1985, Robert Sinchammer, Chancellor of the University of California, Santa Cruz, proposed sequencing the human genome as a grand challenge that could elevate his university's status. He organized a workshop, but the idea didn't gain immediate traction. Then came an unexpected catalyst: the Department of Energy. The DOE had been studying radiation's effects on genes since the atomic bomb's development. They maintained databases on genetic mutations. In 1986, DOE officials proposed sequencing the human genome to better understand radiation damage. This pragmatic justification gave the project credibility. The National Institutes of Health, America's primary biomedical research funder, was initially skeptical. Many prominent scientists opposed the project. They feared it would consume resources better spent on investigator-driven research. David Botstein, a leading geneticist, called it a mindless project. Nobel laureate James Watson himself initially opposed it, arguing that most of the genome was junk DNA, not worth sequencing. But opinions shifted. In 1988, a National Research Council report endorsed the project, calling it feasible and valuable. The same year, James Watson, perhaps history's most famous DNA scientist, reversed his position and agreed to lead the NIH's genome program. Watson's involvement provided crucial credibility and visibility. Watson faced immediate challenges: scientific skepticism, funding competition with other research, and international coordination. The project needed to be international. The human genome belongs to all humanity, not one nation. In 1990, the Human Genome Project officially began as a 15-year, $3 billion effort coordinated by the U.S. through NIH and DOE, with participation from the UK, France, Germany, Japan, and China. The goals were ambitious: sequence the entire 3 billion base pairs, identify all genes estimated at 50,001,000,000, store the data in public databases, and address ethical, legal, and social implications. 5% of the budget was earmarked for ELSI, ethical, legal, and social implications, research. This was unprecedented, building ethical considerations into a scientific megaproject from the beginning. Watson insisted that all data be made public immediately, available to any researcher worldwide at no cost. This Bermuda Principles, established at a 1996 meeting in Bermuda, required that sequence data be released to public databases within 24 hours of generation. This open-access philosophy contrasted with traditional scientific secrecy and would later conflict with commercial interests. The project's first years focused on developing technology and mapping genes locations, identifying landmarks on chromosomes before sequencing the detailed text. Sequencing in 1990 was laborious. The Sanger method, developed in 1977, required radioactive materials, gel electrophoresis, and reading X-ray films. Labs could sequence perhaps 1,000 base pairs daily. At that rate, sequencing 3 billion bases would take 8,000 years. Technology improved rapidly. Automated sequencers replaced manual methods. Fluorescent dyes replaced radioactivity. Capillary sequencers processed samples faster. By the mid-1990s, labs could sequence 10,000 bases daily. Still not fast enough, but progress was accelerating. In 1992, James Watson resigned as director after conflicts with NIH Director Bernadine Healy over patenting gene sequences. Watson opposed patenting. Genes were natural discoveries, not inventions, and should remain in the public domain. Healy supported patenting to maintain American competitiveness. Watson's public criticism of his boss made his position untenable. Francis Collins replaced Watson in 1993. A physician-geneticist who had discovered disease genes including cystic fibrosis, Collins was religious, an evangelical Christian who saw no conflict between faith and science. His leadership would prove crucial through the project's most difficult years. Collins reorganized the effort, focusing on 5 major sequencing centers: the Sanger Center in Britain, now the Wellcome Sanger Institute; Washington University in St. Louis; MIT's Whitehead Institute; Baylor College of Medicine; and the DOE's Joint Genome Institute. These centers would produce the vast majority of sequence data. By the mid-1990s, the project was progressing but slowly. Then, in 1998, a bombshell. Craig Venter, a maverick scientist, announced he would sequence the human genome faster and cheaper than the public project using a controversial technique called shotgun sequencing. Venter was brilliant, brash, and divisive. A Vietnam War medic turned scientist, he'd worked at NIH developing sequencing techniques. Frustrated by bureaucracy and disagreeing with the methodical gene-by-gene approach, Venter left NIH in 1992 to found the Institute for Genomic Research, TIGR. There, he pioneered shotgun sequencing, breaking DNA into small random fragments, sequencing them, then using computers to assemble overlapping pieces like a jigsaw puzzle. The method was controversial. Critics said it wouldn't work for large genomes and would miss repetitive regions. But in 1995, Venter's team used shotgun sequencing to complete the first free-living organism's genome, the bacterium Haemophilus influenzae, 1.8 million base pairs. It worked. In 1998, Venter announced he was founding Celera Genomics, a private company that would sequence the human genome in 3 years for $300 million, a fraction of the public project's cost and time. Venter's announcement shocked the scientific community. Some saw healthy competition spurring progress. Others viewed it as a hostile takeover attempt, privatizing what should be a public good. Venter's business model was ambiguous. He claimed results would be published, but also suggested creating a subscription database charging researchers for access. This violated the Bermuda Principle's open access ethos. The race was on. The public consortium accelerated its timeline, abandoning the careful mapping approach for whole genome shotgun sequencing, the method they'd previously criticized. Collins and Venter became public faces of competing efforts. Media coverage intensified. The genome race made front-page news, rare for basic science. The competition had benefits and costs. It spurred technological innovation and faster progress. Both sides pushed harder than they might have alone, but it also created acrimony, wasted effort on duplication, and threatened the open access principle. Venter's Celera used the public project's freely available data while keeping its own proprietary, benefiting from openness while rejecting reciprocity. This infuriated public project scientists. Behind the scenes, the work was grueling. Sequencing centers ran 24/7. Technicians loaded samples into sequencing machines constantly. Computers assembled millions of sequence fragments. Quality control verified accuracy. The sheer scale was unprecedented. Industrial biology, more factory than traditional lab work. By late 1999, both efforts had substantial data. Rumors circulated about who was ahead. The competition was becoming counterproductive. Ari Patrinos, who ran DOE's genome program, brokered peace talks. The White House got involved. President Clinton saw political value in a joint announcement, showing American scientific leadership and cooperation. After tense negotiations, Collins and Venter agreed to a simultaneous announcement. On June 26, 2000, at the White House ceremony, they jointly declared a working draft of the human genome was complete. Clinton gave his theological framing. Tony Blair appeared by video from London, praising international collaboration. The moment was genuinely historic, if slightly premature. Much work remained. Both groups published their results in February 2001. The public consortium's paper appeared in Nature, Celera's in Science. The findings were revolutionary and surprising. First, humans have far fewer genes than expected. Initial estimates ranged from 50,000 to 100,000 genes. The actual number, around 20,000 to 50,000, not much more than simpler organisms. Roundworms have 20,000 genes. Rice has 40,000. This was humbling. Genetic complexity doesn't equal organism complexity. What matters is how genes are regulated and how proteins interact. Second, only about 2% of the genome codes for proteins. The other 98%, previously dismissed as junk DNA, includes regulatory sequences, structural elements, and regions whose functions remain mysterious. Much junk isn't junk at all but performs important regulatory roles. Third, all humans are 99.9% identical genetically. Race is a social construct with minimal biological basis. Genetic variation within racial groups exceeds variation between them. We're all, genetically speaking, one family. Fourth, humans share genes with all life. About 60% of human genes have counterparts in fruit flies. Nearly 99% are shared with chimpanzees. The genetic differences making us human are surprisingly small, a reminder of our connection to all life on Earth. The draft announcement in 2000 wasn't the end, it was a beginning. The finished sequence, with higher accuracy and fewer gaps, was declared complete in April 2003, 50 years after Watson and Crick's double helix discovery. Even then, some regions remained unsequenced, highly repetitive areas resistant to existing technology. The Human Genome Project's immediate scientific legacy was enormous. It provided a reference sequence researchers could use to identify disease genes, understand genetic variation, and study evolution. The data was freely available online. Anyone, anywhere could access it. This democratization of information accelerated research globally. The technology developed for the project transformed biology. Sequencing costs plummeted. The project cost $3 billion, but by 2022, sequencing a genome costs under $1,000. The $1,000 genome enabled personalized medicine, population studies, and widespread genetic testing. Medical applications were the project's greatest promise. Understanding disease genes could lead to better diagnostics, treatments, and prevention. Some successes materialized quickly. BRCA1 and BRCA2 genes linked to breast and ovarian cancer could be tested. Women testing positive could take preventive measures. Pharmacogenomics, tailoring drug treatments to genetic profiles, emerged. Genetic testing for rare diseases expanded. But translating genome knowledge into medical breakthroughs proved harder than anticipated. Most diseases involve multiple genes and environmental factors. Cancer involves hundreds of genetic changes. Knowing the genome didn't immediately produce cures. The post-genomic era required understanding how genes interact, how they're regulated, and how they respond to environment, vastly more complex than sequencing alone. The ethical implications were profound. The ELSI program addressed issues like genetic discrimination, privacy, patenting, and eugenics. In 2008, the Genetic Information Non-Discrimination Act prohibited discrimination based on genetic information in employment and insurance. But enforcement is imperfect, and concerns persist. Genetic testing raised difficult questions. If you could know your risk for Alzheimer's, Huntington's, or cancer, would you want to? What if there's no cure or prevention? Insurance companies want genetic data to assess risk. Should they have access? Employers might discriminate against high-risk individuals. How do we protect genetic privacy while enabling research? Ancestry testing became popular. Companies like 23andMe and AncestryDNA offered consumer genetic testing. Millions submitted DNA samples, creating massive databases. People discovered unexpected ancestry, unknown relatives, and health risks. But these databases raised privacy concerns, especially after police used genealogy databases to catch criminals like the Golden State Killer. Users uploaded DNA for ancestry, not criminal investigations. The project enabled synthetic biology, designing organisms from scratch. Craig Venter's team created the first synthetic bacterial genome in 2010. This raises possibilities of designing bacteria to produce biofuels, clean pollution, or manufacture medicines. But it also raises concerns about biosecurity and playing God. CRISPR-Cas9, a gene editing technology developed in 2012, allows precise editing of genomes, cutting out disease-causing mutations and inserting correct sequences. It holds promise for curing genetic diseases. In 2019, patients with sickle cell disease and beta thalassemia were treated successfully with CRISPR. But CRISPR also enables germline editing, changing embryos so modifications are inherited. Chinese scientist He Jianqui's 2018 editing of human embryos to confer HIV resistance sparked global outrage. The technology exists, but should we use it? The project revealed human genetic diversity and migration patterns. By comparing genomes from different populations, scientists traced human origins to Africa and mapped ancient migrations. Humans interbred with Neanderthals and Denisovans, extinct human species whose DNA persists in modern humans. We're not pure Homo sapiens but hybrids. This reframes human evolution and identity. The project also demolished scientific racism. Race, as a biological category, is scientifically meaningless. Genetic variation doesn't cluster by continental ancestry in ways that support racial categories. Yet this hasn't eliminated racism. Genetics is sometimes misused to support racial hierarchies. Scientists must combat these misappropriations. The Human Genome Project established models for big science in biology. Particle physics had CERN, astronomy had Hubble Space Telescope. Biology now had genome projects. The approach, Large-scale, collaborative, technology-driven, influenced subsequent efforts. The Cancer Genome Atlas, the Encyclopedia of DNA Elements in Code, and the Human Cell Atlas. The Collins-Venter rivalry had a postscript. Despite their competition, both contributed enormously. Collins remained at NIH, becoming director in 2009 and serving through 2021, leading during Ebola, Zika, and COVID-19 responses. Venter continued genome work, sequenced his own genome, and pursued synthetic biology. They never became friends, but mutual respect developed. The project's completion didn't finish the job. In 2022, scientists finally sequenced the remaining 8% of the genome, the highly repetitive regions previously too difficult to sequence. The truly complete human genome, T2T, telomere to telomere, was only achieved 2 decades after the first draft. Moreover, one genome isn't enough. The Human Genome Project sequenced a composite from multiple donors. Everyone's genome is unique. The Human Genome Diversity Project and 1,000 Genomes Project cataloged genetic variation across populations. Millions of genomes have now been sequenced, revealing the breadth of human diversity. The project also highlighted what we don't understand. Knowing the sequence doesn't mean understanding function. How do 20,000 genes create a human being? How does genotype produce phenotype? How do genes interact with environment? These questions define post-genomic biology. The promise of personalized medicine is partially fulfilled. Genetic testing helps diagnose rare diseases, predict drug responses, and assess cancer risk. But most common diseases, diabetes, heart disease, mental illness, involve complex genetic and environmental interactions not easily addressed with current knowledge. Gene therapy, long promised, is finally delivering. Treatments for inherited blindness, hemophilia, and sickle cell disease have been approved. Cancer immunotherapy, engineering patients' immune cells to fight tumors, has had remarkable successes. These breakthroughs took decades beyond the genome sequence, reminding us that basic knowledge takes time to translate into treatments. The project revealed that nature versus nurture is a false dichotomy. Genes influence traits, but environment matters enormously. Epigenetics, chemical modifications affecting gene expression without changing sequence, shows that experience literally affects gene activity. Trauma, diet, stress, all influence which genes are active. We're not slaves to our genes, nor are genes irrelevant. We're products of complex interactions. The Human Genome Project also democratized science in unexpected ways. Citizen scientists contributed. Fold-It, a protein folding game, recruited gamers to solve problems computers couldn't. Patients with rare diseases formed communities, shared genetic data, and funded research. This participatory model challenges traditional scientific authority. But genomics also risks exacerbating inequality. Wealthy individuals access advanced genetic testing and personalized treatments. Developing countries lack infrastructure for genetic medicine. The benefits of genomic knowledge risk accruing primarily to wealthy nations and individuals, widening health disparities. The philosophical implications are profound. Learning our genetic code raises questions about free will, identity, and determinism. If genes influence behavior, intelligence, and personality, what does that mean for moral responsibility? These questions resist simple answers and require ongoing ethical reflection. From Watson and Crick's 1953 discovery to the 2000 draft announcement, to today's $1,000 genomes and CRISPR editing, genomics has transformed from esoteric science to practical medicine to consumer product. The journey from understanding DNA structure to reading the entire human instruction manual took 50 years. The journey from reading it to truly understanding it continues. The Human Genome Project was biology's moonshot, an audacious goal requiring international collaboration, technological innovation, and massive resources. Like the moon landing, it inspired a generation, demonstrated what collective effort can achieve, and opened possibilities previously unimaginable. Unlike the moon landing, its practical applications are still emerging. We've read the book of life, but we're still learning to comprehend its meaning. When President Clinton declared we were learning the language in which God created life, he captured the project's spiritual dimension. For religious people, reading the genome reveals divine creation's complexity. For secular people, it reveals nature's wonder. Both can agree. We've gained unprecedented insight into what makes us human, what connects us to all life, and what possibilities and responsibilities that knowledge brings. The Human Genome Project's greatest achievement may be humility. We discovered we have barely more genes than worms. We learned we're 98% identical to chimpanzees. We found we're 99.9% identical to each other. We're not as unique, complex, or separate as we imagined. We're related to all life, connected to each other, and far from understanding all the mysteries written in our DNA. That humility should inform how we use genomic knowledge. We have power to read, edit, and potentially redesign genomes. That power demands wisdom, ethics, and caution. The Genome Project built ethics into its foundation. As genomic medicine advances, that ethical framework must evolve too, ensuring benefits are shared, harms are minimized, and human dignity is preserved. From that June day in 2000 when Clinton stood between Collins and Venter to today's routine genome sequencing, the Human Genome Project transformed biology, medicine, and humanity's self-understanding. It was the beginning, not the end, the opening of a book whose chapters we're still writing. The code is known. Now comes the harder work, understanding what it means and using that knowledge wisely.